Welcome and thank you for the opportunity to speak to you today and to share this information. This is the first disclosure in public of the progress made by the Reliance Life Sciences team on the product called Relcovax, which is a vaccine designed to provide protection against SARS-CoV-2 and the associated disease COVID-19. This is a large team that has contributed to the development of this product. Of those participating as authors, we have Dr. Ramana Rao, Fakhar Masand, Reddy, Ramna, and I am very glad to support the team and have been supporting them through the development of this product, which is a second-generation multivalent SARS-CoV-2 vaccine candidate specifically designed to meet global vaccination demands, including for those nations and regions that don't have the economic resources of the Western developed world. I'm going to start with an introduction to Reliance Life Sciences. This is probably the biggest life sciences company that most of you have not heard of before. Reliance is fully owned by promoter companies Reliance Group, which is India's largest private sector enterprise. It has a variety of different businesses, but in the Reliance Life Science Group, it's medical biotechnology based for a variety of products and services, focusing on products for intensive and critical care use in hospitals. The goal of Reliance Life Sciences is to be a global leader through development of cost-effective solutions for a better life. Just providing a brief overview of what Reliance's capabilities are. One moment, I need to minimize that window. It's a wide range, and you can see that there are two new initiatives within the company. That's the Vaccines and Immunology Initiative, which includes both vaccines and neutralizing antibody products, as well as the Innovative Technologies Group that you can see on the right-hand column. As you can see from this example, Reliance is one of the most diverse and highly integrated medical-focused life science technologies globally. Reliance Life Science currently markets its products and services to 65 countries in the world. You can see it's quite diverse geographically in terms of the reach of RLS. Now I'm going to speak about the SARS vaccines currently available in India. As this audience probably knows, infections in India are nearing 33.5 million, with about 450,000 deaths to date. The vaccines approved include both classical spike protein-based advirus vaccines, Covishield, S2, Sputnik 5, RDIF, etc., as well as an inactivated whole variant vaccine called Covaxin from Biotech, and a DNA-based vaccine. This, to my knowledge, is the first DNA vaccine that's been widely deployed. Considering the large population in India which remains to be vaccinated, new vaccines are needed, including ones that are cost-effective and multivalent. Speaking about Relcovax specifically, RLS has really anticipated some of the challenges we're encountering now in vaccinology with SARS-CoV-2 and has focused on developing a two-protein subunit vaccine called Relcovax. The two components are the receptor binding domain from spike, not the holospike protein, as well as the nucleocapsid protein. The adjuvant system is traditional. It's composed of alum and CpG, and the vaccine has the potential to offer strong protection and a better safety profile relative to existing vaccines. The need in India is to develop a strongly immunogenic yet affordable vaccine that's also safe. And what the team at Reliance has developed is a more traditional product using well-established fermentation technology and adjuvants. Focusing now on the immunogenicity studies to support this, The Reliance group has approached this in a open manner with dose ranging beginning with murine models, BALB-C, with either the receptor binding domain plus alt hydrogel at various doses, receptor binding domain plus alt hydrogel plus CpG, or receptor binding domain plus nucleocapsid plus alt hydrogel plus and minus CpG. As you can see, this has all been done in a dose ranging fashion with a prime and then a day 14 boost and a series of subsequent bleeds for serum and then spleen collection for the cellular immunology assays. Moving on to antibody titers, here if in the upper right, you can see the listing of the different groups that I've just reviewed with you and their corresponding titers in terms of antibody titers, geometric mean, anti-receptor binding domain, represented by these three bars. Mindful of the need to minimize dose as much as possible to minimize potential reactogenicity, I suggest that throughout this presentation you focus on group 8, and you can see that we're seeing nice increase in titers throughout the course of the sampling. Focusing now on the nucleocapsid specific IgG titer in serum from days 13, 21, and 28. This has been compressed, so we don't have all of the groups here. They're just kind of creating noise in the data in data analysis. We're focusing on groups 7, 8, and 9. And as I mentioned, you may wish to focus on group 8 throughout much of this presentation. And again, we see nice IgG titers evoked by the combination of adjuvants and antigen. And you can appreciate the focus again on group 10, which is 10 mics of RBD, 10 mics of nucleocapsid plus alt hydrogel plus the CpG adjuvant. Now moving on to ELI spots for interferon gamma. What we note is here these are actually, there's groups of 3 mice. The mouse 2 group is a control group. So we have groups 6, 7, 8, and 9 once again. Pardon me. And these are actually representative samples of ELI spot assays at various levels of cells placed per well. And this is a sample from ends of 3. But again, the data becomes too crowded, so a representative mouse assay has been selected. And again, I suggest focusing on group 8, represented by mouse 8-1. This is the group that has evoked the best antigen-specific T-cell response in mice. So once again, the Reliance team is focused on both cellular and humoral immune responses and on a 2-antigen approach. Moving to micronewts, this is the Wuhan strain. Here again, you can see a comparison of the control group 2 versus group 6 in terms of the neutralization, microneutralization responses, focusing on the blue bars and the vertical axis for percent viral reduction. You can see that we're seeing strong responses with the experimental groups, and particularly with group 8. So this is RT-PCR-based live virus neutralization assay performed by an independent contract research organization, so independent of Reliance Group to minimize bias. And the assay was performed using pooled serums from groups 2, 6, 8, and 9. At a 1 to 640 dilution, all 3 pools showed nearly 60% efficiency in reducing viral replication using this micronewt assay. On the next slide, we're substituting a different endpoint for microneutralization, that's cytopathic effect. And in this case, we're just focusing on the selected group 8. So you're seeing a sample of 12 different animals assayed using CPE as the endpoint. And you can note that all of those serum samples were positive for microneutralization activity, ranging at dilutions from 1 to 160 to greater than 1 to 2560. Of course, the Delta variant neutralization is what's on everybody's mind right now, rather than the parental strain. And so here we see similar plaque reduction neutralization titer assays focusing on the SARS-CoV-2 Delta strain. And once again, please note that in this case, the dose of vaccine is 4 mics for each antigen, as opposed to 10. So this is lower than the selected final dose for group 8. And yet we're still seeing good neutralization for all 6 group serum samples that have been tested. Here's a PRNT assay comparison of the titers elicited by a more parental strain as well as the Delta strain. You can see that there's some reduction in the neutralization activity with the Delta strain compared to the parental comparator. But in this case, note that in group A, you're looking at the neutralizing antibody titers with the 4 mics for both antigen in the blue bars, and with the 10 mics of each antigen plus the adjuvant in the orange bars, that being equivalent. Group 8B is equivalent to group 8. So in summary, concerning the immunogenicity studies, we're observing robust humoral and cellular immune responses against both proteins in mice. 10 mics of both antigens produce sustained titers after boost. The microneutralization responses was demonstrated in an RT-PCR and CPE reduction assay. The titers demonstrated inhibition of both Wuhan and Delta strains with a modest reduction in the Delta PRNT50. Interferon gamma release was observed from immunized splenocytes, indicating a what appears to be a Th1 biased T response, and no antigen-specific IL-17 responses were observed. Using the additional antigen of the nucleocapsid is also associated with an increased T cell response compared to just the receptor binding domain. So group A has been selected going forward. Quickly, just summarizing, the usual panel of preclinical studies have been performed, including antibody production, which we've just reviewed, toxicology in both Worcester rats and New Zealand white rabbits, and a challenge study in golden Syrian hamsters, which is in progress. Results of that study are encouraging, but results are being repeated to ensure we have reproducible results, and we anticipate those will be available in November. Here's a brief overview of the golden hamster study. These are Ns of 6 at low, mid, and high dose, and those analyses, as I mentioned, in comparison with other candidate vaccines or currently marketed products are currently in progress. In terms of the toxicology, no systemic adverse events were seen during the live phase of the studies. Clinical pathology did not indicate any toxicologically relevant changes. 100% seroconversion was observed, and local inflammatory changes typical of traditional vaccine administration were observed at the site of administration, and is consistent with what has previously been observed with alum CpG formulations. The dosing ranged from 5 to 20 mics per protein per animal in both rats and rabbits, and the no adverse event level threshold, upper threshold, was not reached. So no adverse events were observed with any of these doses. In terms of the manufacturing, Reliance has state-of-the-art manufacturing systems that are well characterized and audited regularly, such as isolators and single-use bioreactors. In this case, the product will be manufactured at Plant 2 of Reliance. This is a multi-product GMP manufacturing facility with both microbial and mammalian suites. and it carries out the full range all the way through harvest to fill, finish, and packaging. It's been subjected to audit by numerous national and international regulatory authorities. This is a quick overview of the manufacturing process flow for the receptor binding domain antigen, and you'll note that it employs a mammalian cell culture process, standard process flow. In contrast, the nucleocapsid is manufactured using a microbial fermentation process to keep costs of goods and productivity yield as high as possible. And once again, this drug substance is manufactured with a standard process flow in a fully characterized GMP facility. This is then followed by final formulation of the product and fill finish, in which the two components are formulated and conjugated with the aluminum hydroxide and then formulated with CPG and other excipients, including preservative, in the case of the multi-dose formulation, yielding the final pharmaceutical product. In terms of the clinical development, we're launching very shortly with a phase 1 study involving a prime and boost, boosting on day 28, and testing 3 dose levels in a classical dose escalation routine, staggered with 24 human subjects in each dose arm. The safety evaluation will proceed for 28 days following dose number 2, and we will perform a 14-day bleed to catch difference between primary and recall responses to the vaccine, as well as a 28-day bleed after the second dose. Analyses will include ELISA for anti-RBD and nucleocapsid IgG, plaque reduction neutralization titers against both Delta and Wuhan, and ELISpot and CBA multi-cytokine analysis for T responses. This phase 1 protocol has received authorization to proceed from the Indian regulatory authorities. So we're just wrapping up the final steps before launching that phase 1 trial. In terms of the overall clinical development plan, we're anticipating both a classical phase 2 and separate phase 3 study, focusing on immunogenicity, safety, and efficacy, and expanding to additional populations beyond the healthy normal adult, such as adolescents and the pediatric. We are anticipating and very interested in partnering or otherwise cooperating in a multi-centric, multi-country collaborative study design. In terms of sample sizes, these are powered. We're anticipating about 300 for the phase 2 and 3,000 plus for the phase 3 study, typical of classic vaccine strategy for a well-established vaccine type like this. We anticipate our phase 1 results in the first quarter of 22, and then rolling initially into the phase 2, and then launching phase 3 in a somewhat staggered fashion in Q2 to Q3 of 2022. In addition, we're anticipating additional studies concerning the viral variants as they emerge and a potential booster. And then in closing, I just want to reach out on behalf of the Reliance team. This 2-component vaccine is designed and manufactured entirely at Reliance Life Sciences. It has shown impressive immunogenicity data in animal models, only some of which have been disclosed here. There's other encouraging results that we hope to share in the next month or two. Significant results were obtained for CPE and cytopathic effect and microneutralization assays. Strong plaque reduction neutralization titer data were obtained against both Wuhan and Delta strains. Minimal expected reactions were observed in preclinical studies, suggesting that the reactogenicity should be tolerable in humans. A 90-day single-dose and multi-dose stability study has been completed for these products, and we're about to initiate our phase 1 studies for safety and immunogenicity. As I mentioned at the outset, the key goal here is to have a very competitively priced product that is readily manufactured and suitable for distribution to emerging economies and underserved populations. After the phase 1 is completed, Reliance will be actively looking for partnerships for the clinical trials and potential production agreements. And so I recommend that interested governments and organizations feel free to reach out to Reliance Life Sciences. Our goal is to meet the unmet need for having a competitively priced vaccine suitable for hastening vaccinations across the entire world. With that, I thank you for your time and patience, and look forward to any questions you may have.
Thank you, Dr. Robert, for such an excellent presentation. And this is Dr. Pragya here, and there is no question in the chat box, but I have a question for you. First, I congratulate for coming up with this product, which is definitely needed because a huge number of population in the world is still unvaccinated, and we are looking forward for more vaccine. But as you have presented a very good plan for phase 1, phase 2, and phase 3, which look very nice. Can you, I have two questions. The first, in the phase 3, you are trying to have around 3,000 of the population for trial. What is, how you have decided that number? And second, as you know that in India, more than 80 crore population has been vaccinated, and a huge number of population is infected during the first wave and second wave. So what is the plan to do the efficacy of the vaccine whenever we enter in phase 3?
So speaking ad lib, I haven't consulted with the team about these specific challenges, which you identify and are absolutely correct. As you know, in any outbreak like this, the strategy for large trials can be very challenging. And we now know that natural immunity, that being the immune response provoked by the natural infection, certainly will compromise the ability to gain clear information about the efficacy of this product. My sense is, I'm speaking, as I say, ad lib in response to your question, is that this underscores the need to proceed with the larger trials using a more global cooperative approach that'll be multi-site and multi-country. And there may be better opportunities in Africa, other Asian nations, the Middle East, and Latin America for this. In general, Latin America, for instance, is grossly underserved by vaccine products currently, as is much of Africa. In terms of the target dose of or target sample size of N of 3,000 for the phase 3, this is will be revised based on data from the phase 2 and the endpoints. But in general, regulatory agencies like to apply the rule of 3 and require typically a phase 3 database of exposed persons that would allow detection of an adverse event at the frequency of about 1 in 1,000. Just to recap, 1 in 1,000 based on the latest data from Canada is the adverse event rate for myocarditis in healthier younger adults and adolescents for the RNA vaccine. So the wisdom of classical wisdom of regulatory authorities to focus on an adverse event rate detection of about 1 in 1,000, I think has merit here also, and will allow us to draw some initial conclusions about relative safety. And so it's based on this historic regulatory precedent of the rule of 3 and ability in general statistically to detect rare adverse events that might occur at the rate of about 1 in 1,000. Have I answered your questions, Doctor?
Yes, thank you so much. And we wish all the best for bringing this vaccine as soon as possible. And we wish all the best for good data as well. And thank you so much for participating in today's summit.
It's my great pleasure. And